This is the first of 2 lecture videos I will be recording on protein synthesis. So this first lecture video is on transcription. The second one will be on translation. So for transcription, you need to identify where in the cell transcription occurs, which type of RNA is produced during this process. You need to be able to describe the function of RNA polymerase, the steps in transcription, and finally, you need to be able to describe the process of RNA processing. So this is what we call the central dogma of molecular biology. And what that means is that your DNA is used to make RNA through transcription. RNA is used to make proteins through translation. So remember, it goes from DNA to RNA to protein. So transcription, which is what you will be learning about first, is when the DNA is used to make that messenger RNA. So the process of transcription means to use DNA as a template to make RNA. Okay, so what I wrote on the slide here is transcription copies the information in DNA to produce what we call pre-messenger RNA using base pairing. So you should remember the term base pairing or complementary base pairing from the lecture video on DNA structure and replication. So I'm going to go to the whiteboard to write down some information that you need to know before we proceed. All right. So this information is on transcription. So I'm going to give you a slightly different definition than I had put on the slide, but it all means the same thing. So a strand of DNA, which we call the template or coding strand, is used to synthesize a strand of RNA. And I'm going to add using complementary base pairing rules. Okay. So when I say complementary base pairing rules, I'm going to write a little chart here for you so you can see what I mean. So remember that for DNA, adenine always pairs with thymine. Cytosine always pairs with guanine. They hydrogen bond to connect those 2 strands of DNA. Now it is very similar in transcription. Well, let's put DNA here. Whoops. And I'll put RNA here. Now, remember from the lecture on DNA and biological molecules that there are 3 differences between DNA and RNA. So I want you to take a second and try to remember what those differences are. So when I say take a second, you can pause the video. Now, DNA is double-stranded, RNA is single-stranded. But most importantly, DNA has thymine as a base, but RNA has uracil. And then last, DNA has deoxyribose as its sugar in the nucleotide. RNA has ribose. So remember, DNA and RNA are both nucleic acids, and nucleotides are the building blocks of these nucleic acids. Now, if a DNA has a nucleotide with adenine, It does not pair with thymine because RNA does not have thymine. RNA has uracil. So if there is an adenine in DNA, it base pairs with uracil in RNA. A thymine in DNA pairs with adenine in RNA. Cytosine pairs with guanine, guanine with cytosine. Okay, these are base pairing rules for the manufacturing of RNA from DNA. So let's go back to the slide. Now, here is the table that I made for you on the whiteboard. Now, what you're looking at here in this slide is the actual process of transcription. So these blue strands, I'm going to point to them with this arrow. Those are strands of DNA. That is the DNA double helix. Now, DNA is going to separate in one little area. It's kind of like a transcription bubble. So remember that during DNA replication, the DNA strands fully separated and then daughter strands were made, right? Well, in transcription, the DNA strands will separate in a certain area. RNA will be produced. RNA comes off of the DNA template. The DNA strands go back together to form the DNA double helix. So my arrows are pointing to the DNA strand in the double helix. Now this area, that is the area where the two strands of DNA start to separate. Now, a strand of RNA is being produced from one of the DNA strands. So notice here that I have what is called a template strand of DNA and a non-template strand. I'm not going to get into this in a ton of detail here, but what I do want you to recognize is that only one of the strands of DNA is used to make the RNA. Now, what you are looking at in this picture where it says direction of synthesis, all this means is that RNA nucleotides are being added to make the messenger RNA strand or strand of RNA, right? So let's go over, I'm going to draw a picture here. Actually, let me show you this first. So I want to mention that transcription occurs in the nucleus of the cell. Remember that that is one of the things that I listed as a learning objective. I'm going to write that here for you. So transcription occurs in the nucleus. Okay. Let me move this out of the way here. It doesn't like to move. Okay, so this whole process is occurring in the nucleus of the cell. Let's go over to the whiteboard. I'm going to go to the next page so I have enough room. So here is the beginning of the DNA double helix. Now, I don't think I can draw a curved line. Nope. Okay, so here is the area where the DNA is separating. Okay. So I am going to use the bottom strand as the DNA template strand. So we're going to write DNA template. Okay. Now the one above is just the DNA strand that is not used for transcription. So the DNA double helix separated in this area, and the messenger RNA is going to be transcribed. So let me put a sequence on here, a DNA nucleotide sequence. We'll do A. Okay, so what I'm showing you here is the DNA sequence on the template strand. So now a messenger RNA strand will be created using these complementary base pairing rules. I'm going to draw my messenger RNA. I'm actually going to call it the pre-messenger RNA strand, because it's not technically messenger RNA until RNA processing has occurred. Okay, so now it follows base pairing rules. So adenine does not pair with thymine because there is no thymine in RNA. It pairs with uracil. So we have A pairs with U. Now thymine in DNA pairs with adenine in RNA. So don't get confused here. Don't say, but there is no thymine, right? Because there is still thymine in the DNA. It's just not present in RNA. Okay, so we have T pairs with A, T pairs with A. Guanine always pairs with cytosine. Here we have an adenine in DNA. It pairs with uracil in RNA. Cytosine in DNA pairs with guanine in RNA. And same for this next base. Okay, so what I've shown you here is the synthesis of an RNA strand based on the nucleotide sequence in DNA. Okay. Now there are a couple things I want you to make note of here. And that is transcription is not occurring for that entire DNA strand. Remember that in humans, we have 23 pairs of linear chromosomes. Now, on a chromosome, there are different areas that are your genes. Okay, so if you have a really long chromosome, thousands and thousands and thousands of nucleotides, only a small portion of those nucleotides are actually going to be genes that code for proteins. So transcription occurs for a gene. It is not the entire strand of DNA that is being transcribed. So transcription produces a pre-messenger RNA for a gene on DNA. Okay, not the entire chromosome, not the entire strand of DNA, just the area that is a gene for a protein. Okay, now the slide that I'm going to go back to asks you about the function of RNA polymerase. Now, enzymes end in ASE, so RNA polymerase is an enzyme. Similar to DNA polymerase, RNA polymerase adds nucleotides to the growing strand of RNA. So RNA polymerase adds nucleotides to the growing strand of RNA. Now it will be adding RNA nucleotides, right? They have ribose as their sugar, they still have a phosphate group, and the bases can be adenine, guanine, cytosine, or uracil. Now, once transcription is complete, you have a single strand of RNA. Let's go back here. So this slide shows you RNA polymerase. It is that reddish oval you see in the picture, and it is adding RNA nucleotides to the growing strand of RNA. So DNA polymerase adds DNA nucleotides to a growing strand of DNA. RNA polymerase adds RNA nucleotides to a growing strand of RNA. Now, there are 3 steps to the process of transcription. The 3 steps are initiation, elongation, termination. Now, this is actually a fairly complicated process, but you only need to know the basics. So initiation. To initiate something is to begin, right? So initiation is step 1. RNA polymerase attaches to the DNA strand at an area called the promoter. This is a short sequence, a short nucleotide sequence that the RNA polymerase recognizes as the area where it needs to start transcription. So let me write that down for you. So we're on initiation. So RNA polymerase attaches to the DNA template at the promoter region. So you can see that in the picture I put here. The promoter region is that little yellow area on the DNA double helix. So it kind of like signals the RNA polymerase to attach there. That's the beginning of the gene. That's where it needs to start transcription. Now, elongation is the second step. And all elongation means is that RNA polymerase is adding nucleotides to the growing strand of RNA. Okay, so RNA polymerase adds nucleotides to the growing strand of RNA. Okay. And then finally, we have termination. And you can see here in the picture that everything is being released. Okay, so RNA polymerase detaches from the DNA strand and the new RNA strand is released. And like I said, if you read about this online, and even in your textbook, it goes into a lot more detail, but this is all I really need you to know about this. Initiation is when RNA polymerase sees that promoter region of the DNA, it attaches, and that is where it will start transcription. Elongation, RNA polymerase is adding RNA nucleotides to the growing strand of RNA. I always say, elongation is making it longer, right? Termination, RNA polymerase hits a certain nucleotide sequence in DNA, it dissociates or releases, the RNA strand is released, and transcription ends. Now keep in mind that this all occurs in the nucleus. So just to review what we have gone over so far, The process that we are talking about goes from DNA to RNA to protein. DNA to RNA is transcription. RNA to protein is translation. Transcription occurs in the nucleus. During transcription, a DNA template is used to make a strand of RNA. Okay. And the strand of RNA follows complementary base pairing rules. Adenine in DNA pairs with uracil in RNA. Thymine in DNA pairs with adenine in RNA. And then cytosine and guanine still pair with each other. So if that sounded confusing, go back to my slide where I made the little chart for you. Now, the final part of this video is RNA processing. So I've been using these terms interchangeably: RNA, messenger RNA, pre-messenger RNA. And I want to clarify for you. RNA is like a generic term, right? There are different types of RNA. There's messenger RNA, transfer RNA, ribosomal RNA. Okay, so during transcription, the RNA is called pre-messenger RNA until this processing occurs, then it is messenger RNA. Okay, now RNA processing still occurs in the nucleus, and there are two major steps that you need to remember. The first is that a cap and tail are added to this pre-messenger RNA strand. So let's take a look here. Here is DNA, right? You can see here it says promoter. That is where RNA polymerase will attach at the beginning of transcription. Okay, you get transcription right here, and you end up with pre-messenger RNA. Shows you right here. Now, on this pre-messenger RNA, we have a small segment of nucleotides that we call the cap. It's like this little red region it shows you here, and a sequence of nucleotides that we call the tail. Notice they're all adenine. You don't really need to know why that is, but when you go to read about this from another source, it will probably call it the poly-A tail. That's because it's a bunch of adenines. Okay, but all you need to remember is cap and tail. Those go on either end of that pre-messenger RNA. Okay, now here is the other really important part of RNA processing. So there are two regions in the DNA right here. Now remember, we are coding or we are transcribing a gene, not an entire strand of DNA. It's just a gene. Now, within this gene, there are sections we call exons and sections that are called introns. Exons are what we call the coding regions of DNA. These are the parts of the gene that we actually need to make that protein. So let me show you here. So here, this darker blue area is an exon, right? Actually, I think in this picture, that darker blue area is, yeah, I think that means the exon. So what I want you to notice here on this pre-messenger RNA is here we have an exon. This is an intron. This is an intron, right? This lighter green and medium green areas, those are exons. The messenger RNA needs those exons. Those have to stay put. The introns can be removed. Okay, so the second part of RNA processing is the introns are removed. They are not needed at this point. the exons are all that are needed in that final messenger RNA. So the final result after RNA processing is this. Here you have messenger RNA at the bottom, right there. Okay, it contains the, whoops, erase that. The messenger RNA has the cap, it has the tail, and the exons, which is this portion, this portion, this portion. Now it is what we call messenger RNA. Now, again, sometimes I use RNA, messenger RNA, pre-messenger RNA interchangeably, but they are all slightly different. So let's do a really quick review of transcription. I'm going to go back a couple slides here. Okay, so let's look at this image on the right here. You have DNA, double-stranded DNA, right there. Transcription occurs and RNA processing, and now you have messenger RNA. Messenger RNA is then ready to leave the nucleus and enter the cytoplasm for translation, which is the next lecture video. Okay. So from this video, you need to understand the function of transcription, and that is to produce RNA from a DNA template. Okay. You need to remember complementary base pairing. So here's the chart I gave you. I like this chart. I think it's pretty helpful. You have adenine in DNA pairs with uracil in RNA right here. Thymine in DNA pairs with adenine in RNA, cytosine to guanine, guanine to cytosine. Remember, DNA opens up. The RNA polymerase adds RNA nucleotides to the growing strand of RNA. And then after that is complete, the RNA is released and the RNA polymerase also releases from the DNA. The double helix goes back together, and now you have an RNA strand. Okay. And finally, you need to remember that in RNA processing, a cap and tail are added to the strand after transcription, and those introns are removed. We also call that splicing. So what you end up with is a messenger RNA with a cap, a tail, and exons. And this is for a single gene in DNA.